Hello, I'm Dr. Robert Sizer from Roosevelt University, and in this video I'm going to be talking about vaccine development and testing. This is part of our third module in the series on vaccination science resources. Part 2 of this module is titled From Trials to Vials, and it is going to outline several different areas regarding vaccine development and testing. The first part will be a short review on how vaccines work to help build protective immunity. I'll be talking about the vaccine development pipeline and the process of finding a target for a vaccine, designing a vaccine, subjecting that vaccine candidate to preclinical trials and clinical trials, taking the vaccine candidate through regulatory review and approval. So first, by way of review, if you have not yet watched the first part of this module or the other videos in the series, I highly recommend them. They take you through a background of the SARS-CoV-2 virus and its general biology, as well as some great introductory material on how scientists study anything, but particularly how they study viruses. I'm going to offer a little bit of review here because our ability to understand how vaccines are developed and how they're used is going to depend on our ability to understand how they work and how they stimulate the immune system. So vaccines are used to build protective immunity, or you may have heard the term herd immunity. I talked about it in the previous video. Here, the sense is that vaccination's purpose is to help us have the immune system in effect to protect us against later infections with pathogens. So I'm going to introduce a couple of these terms again. A pathogen is a invasive molecule, protein, cell, something that is recognized as not being part of our body. Here, of course, specifically, we're going to be talking about the SARS-CoV-2 virus as that pathogen. And the infection with this pathogen, just like with any other, leads to a coordinated response by the immune system. The specific recognition of this pathogen is going to lead to a cascade of events, ultimately leading to the development of T cells and B cells, which are specific parts of our immune system that are responsible for coordinating events, generating antibodies, and mounting the cellular response that we need to, again, specifically recognize and target and kill that pathogen. An important part of building protective immunity is the process of immunological memory, which means having cells in our body that are able to recognize and again mount a specific response to that pathogen upon subsequent re-exposure. In other words, to make us prepared for later infections. So our goal, of course, through vaccination is to have this taking place both at the individual level and at the population level. So when it comes to actually using a vaccine, the purpose of that vaccine is to stimulate the immune response for protective immunity without having the risks of disease. In the top part of the figure shown on this slide, someone who receives a vaccine, someone who is immunized and has not previously been exposed to that pathogen, will mount a primary immune response. Their immune system will do the same thing that it will do when it sees the live native pathogen. But instead of potentially mounting a full-blown immune response that could be harmful, could result in sickness or even death, the person will usually have no symptoms. Occasionally they'll have mild symptoms, and that's because in dealing with the vaccine, their immune system is active, but the virus is not replicating. The virus does not spread within the body. And so the person can deal with this essentially milder case. Then on subsequent re-exposure, If that person encounters the live virus by coming in contact with someone who is infected with it, they can mount a secondary immune response. So they're already primed to recognize that pathogen. They know what to do. Their immune system knows what to do when the virus is present in the body, and they're able to again deal with it much more quickly. And typically, those people then will be asymptomatic. They won't get sick, and it's much less likely that they're going to pass it to someone else. So obviously, this is a great goal to have. And you've been hearing a lot about vaccines and their development, and there's been a lot in the news. What I want to do over the most of this video is to walk you through the process of coming up with a vaccine and how we go from looking at the structure of a pathogen, how we look at what happens when that pathogen enters the body, and how the immune system reacts, and how we use that as the basis for an understanding of what might make a good vaccine against that pathogen. And then we'll walk through the process of taking that from that initial set of discoveries and studies all the way to producing it and manufacturing it. And we'll look at some of the things that happen along the way. So these are the basic steps I'll be talking about: exploratory research, preclinical studies, clinical trials, regulatory approval, manufacturing, and quality control. And I call this a pipeline or a timeline, but it isn't. But it's nothing. This isn't a set of things that happens in a truly an exclusively linear way. That is to say that regulatory approval can happen at several different steps in the process. Quality control happens throughout this process. Clinical trials can continue even after approval and after manufacturing continues. So there are a number of different steps that go in a generally chronological order, but there's a lot of things that happen simultaneously as well. So the first step, and this is really in the realm of basic research, is finding a target, finding out what it might be about a pathogen, particularly a virus, that would make it a good candidate for incorporating into a vaccine. So the vaccine itself is going to be a mixture of materials. You need a way of delivering something. You need a way of sort of packaging it up for the body to take in and for the human immune system to respond to. And we need to be able to essentially fool the immune system into being able to take it in and treat it as if it were a live active pathogen, but of course without the harm that live active pathogen can present. The good news in the case of the SARS-CoV-2 virus is that we already had known a fair amount about human coronaviruses and this family of viruses. And we learned pretty quickly about the very specific structure through having genome sequencing for the SARS-CoV-2 virus. That information was available within just a few weeks of the first identification of this virus in the human population in China. And so structural biologists, geneticists, researchers were able to take a look at this information and act on it very quickly. In addition, we had known a fair amount, and we learned a lot rather quickly about the replication cycle of the SARS-CoV-2 and other coronaviruses. And this was based on previous coronavirus outbreaks and other studies that had happened since then, since the MERS outbreak, the SARS outbreak from some years ago. So taken together with this information about what the virus looks like, researchers were able to identify parts of the virus that the immune system would recognize when it was making its response, and how the immune system was able to recognize that virus as being non-self. So that's the big distinction: being able to tell the difference between what things belong to our bodies and what things are outside invading pathogens, and being able to respond accordingly. So we had some good ideas about what a reasonable target or set of targets might be. And what researchers settled on pretty early was the spike proteins, which coat the outside of of the virus. These became the focus of vaccine research, with the idea being that being able to train the immune system to recognize this spike protein antigen would strengthen the body's ability to make neutralizing antibodies and to kill infected cells. So the good thing about using the spike proteins is that they're on the outside of the virus, which means they are accessible and they're recognizable by the components of the immune system, the molecules and cells that make it up. And they're necessary for the virus to enter our cells, so that if we can target them and basically prevent them from doing that, that will not only allow us to recognize the virus, but to prevent it from getting into cells and then replicating and spreading and making more. So spike proteins became an intense target of study, and they turned out to be a pretty good candidate for what would represent this virus in a vaccine. So vaccines are designed, again, to be able to represent a pathogen to the immune system, but without having the risk of having the full-on exposure to the pathogen that can lead to severe illness. Vaccines against that are used to represent viruses are traditionally based on the structure of the virus itself and contain several of its components. The three types of vaccines in use today are so-called conventional, vector-based, and nucleic acid vaccines. Each type has its own pros and cons relating to how it can be produced, how it can be distributed, how good it is at inducing an immune response, how effective and long-lasting that response is, so on and so forth. So the figure in this slide shows a generic virus. It is not necessarily SARS-CoV-2, but it has a general virus structure, and it shows us some of the different things that are used as the basis of vaccines. So these conventional vaccines, for example, would be live attenuated viruses or whole inactivated viruses, where essentially it is the actual virus, but something's been done to it in order to make it less active or to inactivate it entirely so it can't replicate. So you're presenting the full the full virus basically to the immune system, but you're lowering its risk of causing a severe infection. Newer variations on this theme will take just parts of those viruses. They'll mean taking particular viral proteins or viral RNA or DNA or parts of proteins that have been synthesized in the laboratory, finding ways to package those up, present them, introduce them into the body, present them to the immune system, and then hoping to stimulate the same immune response as if the full active virus were present. So conventional vaccines, again, use parts of the virus to trigger an immune response. They might be whole or inactivated proteins or the entire virus. And many early viral vaccines were based on these technologies, and very effective ones as well. The first vaccines were actually small doses of live viruses, which were introduced through a process called variolation. This is no longer used. And one of the key things to note about any vaccine that is produced now is that there is not a risk of having a reaction as if you were being exposed to the live virus. One of the downsides of a conventional vaccine is that it can take a long time to produce in large quantities. So that researchers usually have to have a lot of lead time in order to figure out which form of a vaccine might be useful and be able to produce it and distribute it in the amounts that people would need to use. So in the case of SARS-CoV-2, we've not had that kind of lead time, and so the conventional vaccine candidates that are out there are taking longer to be developed. The figure in this image just shows you how this conventional vaccine production might work. Here's it shows a SARS-CoV-2 clinical strain being grown in the laboratory in a little flask in the upper right. Those are then, those are allowed to infect cells that are growing in that flask. And they reproduce. The virus replicates itself and becomes more numerous. And then that preparation of virus is treated with a chemical that will inactivate it or kill it, so it would not be able to replicate anymore. The inactivated virus would then be purified and then introduced into a vaccine. So once again, the key point is that it is inactive. It is not able to replicate, but it could still stimulate the immune system. Vector-based and nucleic acid vaccines, on the other hand, are newer technologies, but the idea for them has been around for decades. And vector-based vaccines in particular have seen some success before, and nucleic acid-based vaccines are really seeing their major debut with the COVID-19 vaccines that you've been hearing about. These vaccines are designed for safety as well as for specificity. So they look to narrow in on getting the strongest possible immune response with the least possible material that is representing the virus. So they contain only a small set of information about this virus. And in the case of the SARS-CoV-2 vaccines, it is just a single protein, or a portion of a single protein whose information is incorporated into the vaccine. So the vaccine then delivers that viral material so that the host cell can use it to trigger an immune response. And that's what's shown here in the panel in the figure on the right. Here, this is a SARS-CoV-2 infected lung cell, and it doesn't matter whether it's the live virus or the portion of the virus that is being incorporated into the vaccine. The host cell is going to respond in a similar way. It is going to change up its gene expression. It is going to try to make copies of that viral protein based on the information that it's given, and those are going to serve as signals to other cells in the body, particularly to the cells of the immune system, that the cell has become infected. So it's going to mount the same response. That means that antigen-presenting cells, T cells, and then eventually B cells, other cells in the body, are going to be involved in identifying this host cell as having been infected, leading to the death of that cell, and ultimately to the production of antibodies and memory cells of different types that will remember that this pathogen has entered the body. The key distinction, once again, is that when the vaccine is used to do this, it only carries one little bit of information, and that bit of information turns out to be enough to stimulate that immune response, but it is not nearly enough information to be able to make more viruses and infect more cells. So some of our leading non-conventional vaccine candidates, then, the ones that just have some limited set of information about the virus that they're based on, are viral vectors, mRNA vaccines, and DNA vaccines. So all of these three are shown as bringing information about the S protein, or the spike protein, that shows up on the surface of SARS-CoV-2. In the case of the viral vector, it's the DNA that encodes that S protein, and it's placed inside a coat or a package that actually is derived from another type of virus. And this is an adenovirus, one that humans are exposed to all the time. And it basically makes this a delivery vehicle, an effective delivery vehicle, so that we can mount an immune response to it. But we're not going to be making more adenoviruses with S protein DNA into them. The mRNA vaccines I'll talk about in more depth on an upcoming slide. These package up the messenger RNA that encodes the S protein into a lipid nanoparticle. And a DNA vaccine is essentially just the delivery of the DNA that encodes the S protein all by itself. So this is introduced in the body through a process called electroporation, and it's similar to what happens with the viral vector. It's just another way of delivering the DNA into our cells. So a little bit more information on the mRNA vaccine, which is the one that you've heard the most about. These were the first two to be approved and produced and distributed here in the US. So mRNA vaccines, again, encode or include just small pieces of the genetic code for the virus. And these are enclosed or encapsulated into a fatty coat, which is called a lipid nanoparticle. So these are very, very small particles that contain essentially just bits of messenger RNA, which are the template for making the proteins, encoded by a little bit of fat. These particles then are taken up by our cells, and the RNA serves as a template to make a portion of the viral spike protein. So it's this protein then that becomes the pathogen, not the whole virus, but just this bit of the spike protein, which is being produced by our own cellular machinery. And that is enough to stimulate the immune response. So much so that the immune system will then be able to recognize the full protein upon infection with the real virus. So if it says, this is what the spike protein looks like, make some antibodies, let's mount a response to this particular protein. Then when the body recognizes that protein on the surface of an actual virus, it will know what to do. An important note about the RNA vaccine, and with any of these nucleic acid vaccines, is that the RNA inside the particle is not combined with human DNA or with human RNA. It just finds its way into the cells and is used by the same translation machinery, the same parts of our cells that make our own proteins. They just make this little bit extra that happened to come from the virus. There's no indication, and it doesn't happen in our own bodies. It's not going to happen with this bit of RNA from a virus that would be incorporated into our own genome. So it won't be combined with human DNA. All right, so having identified this candidate and having come up with a way to deliver this bit of the viral genome into our cells, the next step is to actually try out this vaccine, try out this way of making something that our immune systems can recognize and help us have protective immunity. So there's a whole series of studies that need to happen. And you've probably heard a lot about clinical trials, but there are a number of things that take place before clinical trials are even started. And these are called preclinical studies. So these are basically lab experiments in which researchers are working with cell cultures. They are growing human cells in the laboratory. They are infecting them with the virus. They are also using the vaccines first to find out what happens to those cells. They may also do some studies with animals that are susceptible to the virus, or with what other pathogen is being studied, to see whether the vaccines will give a protective advantage and will stimulate immunity in those animals. So this happens before any tests are conducted on people. The purpose of these questions, or purpose of these preclinical studies, is to ask, does the vaccine have the desired effect? Does it lead to an immune response? And do researchers have the procedures to make, test, and administer this vaccine properly? So it helps us understand how much of the vaccine we might need, how it should be given, how it should be packaged, etc. The next step is actually a series of steps, and that is to embark on clinical trials. Clinical trials are experiments that are done to test the efficacy of a vaccine, just like with any other type of drug or treatment, and it uses human subjects. So there are several different phases of clinical trials. The first is called phase 1, and it usually involves a small number, just a few dozen healthy volunteers. And the purpose of these studies is just to be able to give the vaccine to those volunteers and ask, is it safe to administer? Are there any serious side effects? Does anybody get really sick from just getting the vaccine? Typically, these can be conducted rather quickly before moving on to phase 2, which is slightly larger in scope. It can contain hundreds of volunteers, and some of those volunteers will be people at high risk. Now, in other clinical trials, that might be people who have a condition that the treatment is intended to treat. In the case of vaccination, it's people who would be at high risk of getting infected and getting sick with COVID-19. So that can include the elderly, people who work in healthcare settings, et cetera, people who are at higher risk of contracting it. So the questions with a phase 2 trial are, how do participants' immune systems respond to vaccination? Do they actually start making antibodies? Do they get protective immunity? What evidence do we have that this is happening? And what are common reactions to immunization? So phase 1 is to ask, is there anything that goes seriously wrong when people are inoculated with this vaccine? Phase 2 is about asking, what is the most likely response upon vaccination? Do they have mild symptoms? Do they have more moderate symptoms? What can we reasonably expect from people? So they watch these cases very closely, because if it looks like people are getting protective immunity and they do not have strong reactions to the immunization process itself, they want to go as quickly as possible into phase 3 trials. So phase 3 will involve thousands of volunteers from diverse groups of people, low risk, high risk, younger, older, et cetera. And these tests will tend to take place at multiple sites, multiple locations. So both of the trials that you may have heard about for the mRNA vaccines were involving upwards of 30, 40,000 people in multiple countries. So this is to get the fullest possible picture of the likelihood of being able to get protective immunity and ultimately herd immunity through vaccination. So the questions involved are, how do vaccinated and non-vaccinated people respond to viral exposure? What procedures and doses should be used? How long does immunity last? And how do people react to the vaccine based on their age or their race or other factors? So that's why you need a lot of people, because you want to have a large number in each of the categories. And importantly, you need to have people who are both receiving the vaccine and receiving a placebo. So you may have heard that researchers and lots of other people were very excited about the outcomes of the clinical trials, the phase 3 trials, for both of the major mRNA vaccines under development. These were developed by a company called Moderna and another company called Pfizer, working with a company called BioNTech. So these are the first mRNA vaccines, and the figure on the right shows you some of the data from one of the trials. This is from the Pfizer BioNTech trial. And it shows us what's called an incidence curve. It shows us how many people got COVID-19 after being inoculated, after being immunized with the mRNA vaccine that they were developing. So the line in blue, or the curve in blue, shows the number of people who got COVID-19 after getting the vaccine dose. And the red dots show us the cumulative number of people over time who got COVID-19 and did not receive the vaccine. They received a placebo. So you can see that after about 7 to 10 days of similar looking curves, all of a sudden the blue line flattens out. So people who had been vaccinated, after about 10 days, they stopped getting COVID-19. And the number only goes up very slightly. You can you'd be able to see that the numbers go out to about 112 days, so about 3 months. Whereas people continued to get COVID-19 at some fairly standard, very steady rate over that same period of time. So this was pretty exciting results, that the vaccine was very effective. People who got the vaccine did not get COVID-19. And for both of those mRNA vaccine candidates, the phase 3 trials reported over 90% efficacy, which means that more than 90% of the people who got the vaccine were protected and did not get COVID-19 during the trial period. So this is considered very strong evidence that the vaccine, both of these vaccines, which saw similar results, were able to confer protective immunity, and that immunity was going to last over at least several months. Now we know that it should be at least 5 or 6 months, based on how these trials have progressed and how they followed people through those trials. There are other vaccines that are currently in phase 3 trials, or in phase 1 or in phase 2. One that I'll mention here is the Oxford AstraZeneca vaccine, which is the leading adenoviral vector vaccine. So another one of these sort of newer generation vaccine candidates. All right, so researchers make vaccines, they're able to test them, they take them through clinical trials, and again, all along the way, they need to have regulatory approvals. That is, somebody who needs to say, looks like you're following the right procedures, you're doing this safely, you're making sure that you're taking care of the people who are signed up for your studies, you're monitoring them, and you're protecting their information. It's going to be safe to produce this vaccine and distribute it for the general public. So all of those things have to be assured before the vaccine can come to market. So there's a number of different things along the way. Many of these are overseen by the U.S. Food and Drug Administration or by their counterparts in other countries. In the U.S., that will include things like investigational new drug applications to let the FDA know that you want to develop this drug or vaccine, pre-licensure clinical trials. So this is the preclinical trials or studies that I talked about before. Presentation of the findings and discussion by the FDA's Vaccines and Related Biological Products Advisory Committee, VRBPAC, which is why we use the acronym. And then biologics license application and inspection of manufacturing facilities and usability testing and product labeling, all sorts of different things along the way. Both of the mRNA vaccines that are now in use in the US have gone through this process and have gotten what's called an EUA, or emergency use authorization, which basically fast tracks them for production. It puts them at the top of the list. And in fact, for all of these candidates, no steps are being skipped, but most of them are going faster than they normally would because, of course, COVID-19 vaccines are top approval priorities. We want to be able to get as many different types of vaccines that are effective out to people as quickly as possible. So companies are continuing to gather trials data and are securing approval to make and distribute vaccines in large quantities. And in just looking back at over the past year, it has been really remarkable, the rate of development and approval. There are many things that are really disturbing and concerning and tragic about the COVID-19 pandemic. One of the things that has gone well and that we should not lose sight of is the fact that we were able to come up with vaccine candidates that work very well. And this process happened in less than a year, whereas typically it has been at least 3 to 5 years for any other comparable sort of vaccine development. So that means that vaccines are being distributed, and that they work. And we expect that widespread vaccine availability can't come soon enough, but is expected by the summer of this year, 2021. All right, so the one sentence summary of this whole talk, the take home message for you is that scientists have the tools and the knowledge to develop safe and effective COVID-19 vaccines. We've seen how they've used information taken from basic research and our fundamental understandings of how the virus infects our bodies, how our immune system responds to the virus, and lots of other things that we've now learned about the molecular tools that we can use to make viruses, excuse me, to make vaccines. And we've been able to develop some vaccines and have others in the development process that are likely to be effective and to be able to be used in a number of different ways. So we're really, really optimistic about having that ability to deal not only with this pathogen, but with other similar threats in the future. All right. So thanks very much for watching. In the last video in this module, I will be talking about the vaccine distribution and allocation process, and some of the challenges and opportunities that presents as well. Thanks very much for watching.